What are the strength and limitation of the DGE? So the key strength of the DGGE is our band excision enable direct identification. So you can have multiple band like 1, 2, 3, 4, 5 bands. So band, you just take out the bands on the gel, maybe on agarose gel or maybe on page polyacrylamide gel electrophoresis. It depends on what kind of a size of the DNA you are looking into. So band excision enable direct identification of the marker, then accident resolution of dominant species. So you can resolve the multiple strains within the species. Say for example, E. coli has multiple strains and subtype based on these signature sequences. You can you can separate them easily. Then visual interpretation of community patterns. So just on the agarose gel patterns or page gel patterns, you can visualize the variation among these genome. Reproducible and reliable results. So your results are very, very reproducible. But once you identify the markers, and this marker can be reproducible with the same primers, if you can identify through PCR and then sequencing, or through some other mode of method. Cost effective for routine analysis. You can routinely do it, just looking at the same standardized condition of DGGE, and you can identify these markers and you can find variation. For example, if you find strain E. coli, strain E. coli 1. Now, how strain E. coli 1 is different from strain E. coli 2? So this can be identified through this marker. So this marker. Now, this marker are basically nothing but identified from the same DGGE. All right. Now, so this is the strength. So cost-effective routine analysis can be done. Now, notable limitations iski kya hai? What are the notable limitations? Now, the complex and band patterns may be overlapped. Sometimes what happens, these band patterns gel. So, suppose a lot of bands are being overlapped here. Now, these overlapped bands belong to the same size, same patterns. So, sometimes it is very difficult to look into the overlapped band pattern because of the similar size, although they are different. So, there might be, for example, A, T, G, C, 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 for example. This is one sequence having this band. Then you can have A, T, C, C, G, G, another band. Now, both the size is same. They should come on the same pattern, same size, but the sequences from here, see, is different. From a single sequence difference, you find variation, or we call the mutant. This is the topic. This is the limitations. So complex band patterns may overlap because of the mutations. Remain intact the size of the sequences. Less sensitive to rare texture detection. So rare texture detection are difficult for the reason because for many reasons. For example, sometimes for the rare sequences are not available. Sometimes if you want to compare with the databases, the rare sequence data is not available. So these are some reasons where it might have less sensitive for prediction. Requires optimization for different sample. So need to have it's not like that you have one sample, 2 sample, 3 sample and you can optimize it. No, you need to have multiple steps to carry out the different samples to identify, and this is how this is one of the limitations. Then again, you have a time intensive gel preparation, so you have to have a lot of gel preparations taking place and so on. Now then come to the TGGE. This is the second method. Now TGGE is basically temperature gradient gel electrophoresis. Now in case of temperature gradient gel electrophoresis, you have exactly the same process as we talked about in DGGE. In the DGGE, you have the chemical or denaturing agent which is used in order to denature these sequences in the gradient mode, from the top to bottom or from bottom to top, from high to low or from low to high. Here in this case, similar work is being done by the temperature. So you have to prepare the gel with the temperature gradient, high temperature, low temperature, low temperature, low, 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 like this. So gradients in the temperature will again provide a certain precise point variation in the two different genome right, to denature. So this is what the purpose. The whole principle of this is based on the same. So once you differentiate these two, then you can easily differentiate on the banding pattern. And this banding pattern is basically will tell you, okay, this is one type, this is second type, this is third type, and so on. So temperature gradient means uses controlled temperature difference, like in gradients, rather than the chemical denaturation. Just like in other, we have taken that chemical. Here we take the chemical temperature. Melting behavior. Now, DNA fragments separate based on their specific melting temperature. And what is the basis of melting temperature? The basis of melting temperature is the intrinsic character of the genome. This is what we are trying to find out. GC content. High GC content, high melting temperature. Low GC content, low melting temperature. So we have to look into melting temperature. Then microbial ecology. What is microbial ecology? Frequently used interchangeably with TGGE, environmental microbiology study. So you can have a different ecology, which microbial ecology, which microorganisms you are using in order to for that DGGE. Now, if you compare TGGE versus DGGE, as I've told, principle is same, method is different, mode is different. Here, in one you have a chemical, in another you have a temperature. So if you compare these two together, you find TGGE is a very simple gel, simpler gel, and DGGE is sort of more stable gradient. So this is the benefit. Why it is more stable? Because you have a complete gradients of a not temperature but the chemical, which denatured regularly the DNA and make a difference. Suppose one DNA come at this temperature, the other DNA is also coming on the same temperature, but this DNA get denatured, this DNA is not denatured. So based on the denaturing and non-denaturing condition, you can have a variation in the banding patterns, mobility patterns, and so you can identify this is one type, this is two type. Then no chemical denaturing handling. This is one other advantage of this. But in this case of one of the advantages is that higher reproducibility. Then you have direct temperature control in this case. In this case, you have a better stabilized, established protocols are available because all the chemicals they have standard concentrations to be used, so you can use any protocol. But in case of temperature, you need to have optimized lot of time is consuming optimization of the temperature. Then reduced chemical waste in this case. Here, wider literature support. So you have a lot of literatures available. So this is some benefits and advantages of the TGE and DGGE in terms of like use of these techniques in your own lab, in your own course, on your own practical side. But both methods provide the community fingerprinting and enable band sequencing for phylogenetic identification. So making differentiation, mutations, variations in these two just by differentiating with the banding patterns of this. Now, TGGE application in environmental microbiology is commonly used as a ecosystem monitoring. So, tracking the microbial community, dynamic in natural ecosystem, including forest, wetlands, and marine environment, right? Whereas temporal studies, you have monitoring seasonal and long-term changes in the microbial community because over a period of time, they keep changing. So when they keep changing, you can identify the changes through these marker methods of DGGE and TGE method. Global research widely adopted the environmental microbiology or ecology laboratories worldwide for comprehensive diversity comparison. So look into the diversity comparison in the most microbiology and ecological labs where you can differentiate these two individuals through these methods. They are very commonly used throughout the globe. Now we have another topic start here. So in my second lecture, so this is my second lecture where we will be talking on understanding of ARDRA and ARISA. In earlier talk, we talk about the DGGE and TGGE. Here we talk about the ARDRA and ARISA. Now, ARDRA is a new topic, although it is also used for comparing the variation or the mutation among the microorganisms through different modes. So again, we can call these two are the markers, but these markers are based on certain different principle and different methods. So we'll discuss what are the different methods and different principles based on these markers. The ARDRA method by name is called as amplified rDNA restriction analysis combined with the PCR amplification of the ribosomal DNA with the restriction enzyme, the addition, to generate the characteristic fragment pattern. Now, if you go my just the earlier lecture where we talk about the DGGE and TGGE, that is the gradient and temperature gradient electrophoresis and denaturing gel gradient electrophoresis, the basic principle that the variation in patterns of these band is same as that here. So in this case also, method is different, but outcome is same. What is the basis of visualizing the difference is same. For example, again, I can make the same gel here, right? And you have a different, this is gel. So this is one pattern. So we call them as a uniform pattern. So 1, 2, 3, 4 uniform pattern. Now, in another case, if you look into like this, so this is a non-uniform pattern. Now, this is the uniform pattern, this is the non-uniform pattern. Now, in non-uniform pattern means this band varies from this in their mobility. Mobility, why? It different in mobility because it varies in terms of like denaturing conditions, or earlier case, or here in terms of like a size. It may be 1 kb, it may be 1.5 kb, it could be plus minus. So depending upon the size, smaller is the size, SS, smaller is the size, faster is a run, FR, faster is a run, then larger is the size, LS, slow is a run, SR. So this is basically the basic principle behind this technique. Now what is happening here in ARDRA, markers are, I call it marker as well as a technique, the target rDNA sequencing, our target rDNA sequencing was amplified. You have to use a specific restriction enzyme. So once you get a sequence, use a specific restriction enzyme, and these, for example, you have say for example here you have, this is one fragment, this is another fragment, this is another fragment. So this are the fragment from the same individual. You have rDNA sequencing. Now in case of 1, 3, so a specific restriction, for example here we are using ECO-R1. This ECO-R1 sequencing are here. In this case, this ECO-R1 sequence is get mutated and it either shift here or it is lost. In both the cases, you can have it will cut like this and this, and if it shift from here to here, then it can have cut like this. So see the difference. This is 1, this is 2, this is size of the fragment. See this side of fragment, this and this. So the variation in the size of the fragment due to the change in the mutation, identifying a specific restriction site, give you different size and so different patterns of the banding. So this is very precise, very accurate, and very advanced techniques. So we call amplified rDNA restriction analysis. Means you have to have amplification of the rDNA sequence or whatever size. Then you have to identify certain restriction site to choose to in order to cut it, and based on the variation in the genetic genome pattern or the gene pattern, the size will be varying. I think it's clear to you. Then create a fragment length polymorphism. So this is that variation. This is called polymorphism. One band is large and another band is small, then you have a large, a small, so on. So the variation in the band pattern reflects the phylogenetic changes. You can visualize on agarose gel or page. Now, in case of difference between page and agarose, so suppose if I ask you a question, when do you run a page and when do you run a agarose? Agarose versus page. Agarose versus page means agarose can resolve large size and they can small size, but there's a limit of large size and a small size. The small size limit I can go, large size limit has no limit. It can go for 10 kb, 20 kb, even whole genome and all. In this small size, what is the resolving power of the agarose gel and resolving power of the page is important to understand. The resolving power of agarose, so we call it resolving power. Now, resolving power means visualization limit. What you can suppose if you have this hand like this, so it looks like you have two hands together. It's like a single hand. But if we separate that hand is like this small one, you can say, okay, these are the two. As long as they are not separated, it is very difficult to visualize them as their two hands are complex of a one hand. Similarly, when the two bands of equal size get on the overlapping conditions, in the overlapping condition, it looks like a single band, refer to the single type of genome, but it is not. So suppose you have a single mutation, and due to this single mutation, there's no variation. Either you deletion or insertion or some changes. Changes may also affect the denaturation. For example, if you change AT to GC, it will affect, right? Or if you change otherwise GC to AT, it will affect, right, due to melting denaturation. Now, what is the smallest limit for the page polyacrylamide gel electrophoresis is the single SNP, single nucleotide polymorphism, right? Even a mutation variation in the single nucleotide will be catched by the page through this method, and that's how the sequencing is done on the page, through page. Now, ERISA enhancement we call automated ribosomal intergenic spacer analysis focused on the intergenic spacer region of the enhanced resolution and automated processing. Similarly, ARDRA method, ERISA is also one of the methods where we can use this powerful tool in order to understand the identifying different markers. In case of this, again the workflow, the processing of this ARDRA or ARISA is the PCR amplification of ribosomal DNA or intergenic spacer. Now, what is the intergenic spacer? So this is the intergenic spacer like this. So RNA sequences are ribosomal RNA sequences. They have intergenic region, IGR. This intergenic region is basically the conserved regions, right? So just like rDNA, you can also interspacer regions or intergenic regions can also be used for the identification of the species. So every species has a very conserved interspacer regions. Interspacer regions are smaller. They can run on the agarose gel easily and can identify it. The ribosomal DNA are intergenic spacer or space region amplified using the specific primer set. Now, the uniform or we call the universal primer sets. So these primer sets are available and you can use these in universal primer set to amplify these interspacer region or rDNA sequencing or rDNA region. Once you amplify these rDNA sequencing sequences region or interspace region, then you can perform the PCR using the primers, and this PCR product When amplified, we call it amplicon. Now this amplicon is again used to electrophoresis. On the electrophoresis, you can, when you run the amplicon, they give you the different banding patterns. And these banding patterns are basically nothing but they are the patterns where you can show the variation in the banding pattern due to the difference in the size of these bands. Now, why these sizes are different? These sizes are different because of the mutation. So, same individuals, same category, E. coli, E. coli, E. coli, but they are different strain. Having variation, changes in the size of the genome, mutations are changed, so on and so forth, through these markers can be identified, and you will end up with the variation. So, this variation can be detected through these ARDRA and ERISA method. Then after this, you have to look into the patterns, the fragment patterns compared and analyzed for the microbial community assessment. As I've told you in earlier slides, like this, here you show the banding patterns, 1, 2, 3, 3 different types of banding patterns. Similarly, you can see here in this method, right, ERISA and ARDRA. So, you amplified rDNA restriction analysis. This is called automated ribosomal intergenic spacer. So, the only difference is that here we are using, in ERISA we are using the interspace region, and ARDRA we are using the same rDNA sequence, but be digested with the restriction enzyme. Now, you can ask a question, single restriction enzyme is used for digestion, give you different pattern. Yes, this is the beauty of this technique. Single restriction enzyme, if you are not using single restriction enzyme, you will mess up the whole experiment. You cannot have, find a variation in these two, and you cannot have any rule out the possibility of the whether they belong to the same or they belong to the different. Now, ARDRA, ERISA research applications, what are the applications of this? Of course, application is the strain differentiation. What is a strain? A strain is something beyond the species. So, species is, I call them as a species like E. coli. So, coli is a species. Under the Escherichia, you have a number of species, but the single Escherichia coli species may have thousands and thousands of different strains. Now, these thousands and thousands of different strains differentiate through this method, through ARDRA, through ERISA, through DGGE, TGGE methods. So, differentiating or distinguishing closely related bacteria. Now, what we call closely related means within the E. coli, you can have a multiple bacteria of the same species as same genera. So, very closely related. So, on the phylogenetic tree, if you look onto the branches, you find on the same tree, same branch, you find the E. coli or Pseudomonas or Klebsiella. And these E. coli, Klebsiella or Pseudomonas, they are very easily detected if they varies in terms of like the mutations and changes through this ARDRA and ERISA method. So, strain differentiation among the related bacterial species, right, with high precision, essential for the taxonomic and epidemiological study. Now, what is epidemiological studies? Epidemiological studies means if you go back in 2019, 2020, 21 and so on, there is a COVID pandemic. Now, in the COVID pandemic, you find the different variants, different Omicron, Omega and so on and so forth. Now, these 3, 4, 5 different variants of the SARS-CoV-2 virus were found to be epidemic in a particular region. For example, Omicron is epidemic in India, Italy and on. Similarly, epidemiology is a study where you identified a specific strain of a species belong causing a certain disease or infection or breakthrough, right, or outbreak. And this outbreak is basically responsible for some disease, some infection from a single strain identified by this method. So, this is called epidemiological significance or taxonomic significance. Then we have environmental assessment. In environmental assessment, comprehensive microbial diversity. It's well known. Analysis in soil, compost system and aquatic environment. All these environmental conditions can be identified in their variation through these methods. complementary analysis, work synergistically with DGGE and TGGE methods for complete community structure characterization. So you can also make cross-check between ARDRA and ERISA for basically comparing the two other methods we call DGGE and TGGE for this purpose. Now, what are the advantages and challenges of these two methods? The key strength is high throughput capabilities for large sample sets. So you can have, large sample sets can be easily identified through this method. Relatively simple methodology and equipment, excellent for strain level identification, cost effective for routine screening, and automated processing available. Now, though these are the strength, but I think one thing I would like to share with you is the skilled requirement. This technique requires a lot of skills because you need to have the designing primers, and then you have to have the sequencing, and then you have to have restriction digestions and to run a gel. So it need a good practice and good high skill development for a technical background. What are the challenges? See, the required careful selection of a restriction digestion, which restriction enzyme you are using, which size of the cutter you are using, 4 cutter, 6 cutters, 8 cutters. This is again very important. So usually 4 cutters are used for that. Limited phylogenetic resolution compared to the sequencing. Because if you compare the whole sequence or the whole genome sequencing or the whole metagenome sequencing, that's more advantageous. This has a limited scope of identifying because it is very small sequences. It could have variations in some other sequences as well, which you're not touching upon. It may need multiple enzymes for complete analysis. So you have 1 enzyme, 2 enzymes, 3 enzymes. So multiple enzymes are required. So again, every enzyme you need to have kind of a, every time you have to set the reactions and do the reactions. And if you lost one reaction, then your results will be lost. Now, what is the workflow? Basically, workflow for that is the same as the sequencing, PCR, and then running the gel after the digestion. Now, compared to that, if you talk about the T-RFLP, we call terminal restriction fragment length polymorphism. This is the very old technique we call RFLP, restriction fragment length polymorphism. Now, what is restriction fragment length polymorphism? It is the polymorphism of the different, for example, this is 3 sequences, 1, 2, 3. Now, in these 3 sequences, you have like this. Now, if you cut these at 3, is this side, and this side, this side, this side, this side, and this side. Now, in these 3 sequences, they are same sequences, but variation in the restriction site. Now, their size is same, the sequence is same, but the restriction digestion size have different positions due to mutation, due to variation. This can be detected through RFLP. You just cut 1, 2, 3, you got 1, 2, 3, 4 bands, you got 3 bands, you got 3 bands. And these 3 bands of this, 3 bands of this might be variation in the size that can be detected, identified on the gel or bending pattern. But this technique we call terminal restriction fragment length polymorphism is fluorescence based techniques, more precise, more highly detectable, and very precisely a very good technique, highly sensitive technique. What is done in this technique? The fluorescent PCR is done. So you have a fluorescent probe to be attached. So first PCR amplification using fluorescently labeled primers used. So when you use a primer, for example, this is bind, use a primer here. Now this primer is a fluorescently tagged. Now this fluorescently tagged primer bind and it amplify. So when the amplicon is formed and you digest it, you find a fragment can be detected through fluorescence microscopy, through fluorescence dye, through different fluorescence techniques. So this is the advantage, the fluorescence tag is remain attached. Then you can cut with the restriction digestion 1, 2, 3. So you will get different fragments. So the terminal fragment you identified of different size. Sometimes, say for example, Suppose if you have okay, suppose if you have this fragment, this fragment, this fragment, you have primer design here and you amplified it. Now here is a tag 1, 2, 2, 3. Now when you cut with the resection digestion, one fragment is got of this size, one got this size, one got this size. So now you got a 3 fragment. One fragment is coming this size, then you have this size fragment, then you can have this size fragment. So first fragment is basically 2. So when you run these 3 fragment on the gel, you find variation in the bending patterns. This is smallest, and that can be easily in earlier case like resection fragment length polymorphism, or in other techniques we are not using fluorescence probe. So we have to look into the visualization through naked eye, and this visualizing through naked eye sometimes fail to identify. But here you need a fluorescent microscope, you need fluorescent technique to identify the fluorescence probe attached here. That can easily, it's a very sensitive technique. So pattern is same, principle is same, but the technique is very sensitive. Enzyme detection, enzyme digestions produce terminal fragment varying length. See, एक is one length, second length, third length. Then you electrophoresed on electrophoresis and you find the difference. So DNA sequencing generate, you can also get a sequence 1, 2, 3. Another technique. So when you sequence this fragment, you find the different peaks. So you find different peaks like this. You don't have a different color of pan, but you find this kind of a peaks. And these peaks can be easily A, T, C, 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 so on. So this is how you can identify these peaks. Now, fluorescence help to detect easily, and this DNA sequence generate a peak representing the community members 1, 2, 3, 4, 5, 6, and so on. So within the species, number of members identified through these sensitive techniques. Now, T-RFLP method overview. Fluorescence primer PCR amplification I have just explained. Restriction enzyme digestion, next step. Then capillary electrophoresis separation is one thing. Then also you can go for the sequencing, automated detection and analysis, and electrophorograms, that is the electrophoresis that you done. So the gel of electrophoresis can be checked into the gel dock or chemidoc or densitometer, whatever instrument you have, you can check it, and then you can interpret based on the banding patterns and the variation in the banding patterns. Now, what is the outcome of this technique process? We call data output. So the method produce detailed electrophoregram with the quantitative information about the microbial community, compositions, and relative abundance. Now, I give you the like this. Suppose if you run 3 fragment, 1, 2, 3, 4 fragment. Now these 3, 4 fragments are basically of different size, so they will come at different patterns. For example, like this. So this is called the electrophoregram. This electrophoregram will give you the clear-cut picture what these 3 bands are and belong to. Okay. Now the T-RFLP method overview means like you have to look into the what are the overview of this method. Okay, I have already explained in detail, so this is just like overview, and this is the data outcome. So this is the data outcome in terms of like a banding patterns, right? Terminal restriction fragment length polymorphism data interpretation, like you can have x-axis then y-axis. So in x-axis you have a fluorescence size measured like this, x-axis, the y-axis. So in x-axis you have a fragment size measured in the base pair, 10, 20, 30, 40, 50, indicating molecular weight differences, size of the 10 kb, 8 kb, 2 kb, 1 kb, 800 base pairs, and so on and so forth. On y-axis you have a fluorescence intensity like this, so like this, so intensity, and which is a relative abundance of community members. Okay, and then peak analysis. So these peaks were analyzed, how intense the peaks are, right? What is the size of these peaks? Where does the peak comes down? This can be identified. So usually people do through sequencing, and the sequencing method can easily identify that. But also through fluorescence techniques also we can use these peaks. What are the applications of these with the case studies we can explain. So TFLP are terminal fragment, terminal restriction fragment length polymorphism, or we also call it fragment length polymorphism, is basically a lot of applications. One is spatial monitoring. So tracking the bacterial community variation, that is changes, variations, mutations, both in viruses and bacteria across the different ecosystem, different niches like soil locations, agricultures, then you have water bodies and clinical samples and so on. And that also has importance of the clinical epidemiology in medical sciences. In agriculture, you can compare the transgenic plant with the non-transgenic plant, change in mutated plants and so on, and variation in types of crops, quality of crops. Suppose if you generate a disease-resistant crop with certain markers can be checked with this method. Then temporal changes, for example, monitoring seasonals and treatment-induced shift. So how the treatment and monitoring the seasonal and treatment-induced means during seasonal and treatment, you can change the physiology of the individuals. Could be bacteria, could be fungus and plant also, which can shift the microbial population over time due to variation, due to changes. That can be checked also from different timeline, longitudinal study, different time scale, different regional, and so on and so forth. The clinical application is that look into the epidemiology, environmental, agriculture, clinical microbiology application worldwide to look into the epidemic mode or epidemic nature of these microorganisms through these techniques. That's the best method to look into the epidemiology. Now, T-RFLP advantages and limitations we talk about. In earlier cases also similarly you have a fully automated high throughput processing, quantitative relative abundance measurement, then high reproducibility, sensitivity, stable, highly stable for complex community analysis. For example, if you have a environmental different community, for example, water bodies, sewage water, hospital sewage water, domestic sewage water, you can look into a different time periods, longitudinal studies, and find the variations through RFLP remain very stable. Statistical analysis is compatibility, so you can also statistically signify, significance can be checked. Statistical analysis can be done on that. Now, what are the limitations which may sometimes make the scientists not to do this technique or get rid of this technique and try to think something else? The different taxa may share identical fragment size. So sometimes it happens there's a lot of overlapping of these taxa size, genome size. That is where we can have to differentiate with a different technique. Now, peak overlap, just like a band overlap in the case of gel. Then multiple enzymes often require for resolution. So not like earlier, you can have multiple enzymes may also. So for example, you need to have one enzyme or two enzymes or three enzymes of different catalysts can be identified. Then you can requires expensive sequencing equipment also. Costs around 1 crore or more than 2 crores equipment, depending upon what kind of a sequencer equipment you have. In spite of or despite of the limitation of these, this terminal fragment length polymorphism remain a very powerful tool, very sensitive and very good tools, though it is very expensive. But from research point of view, it's very sensitive and very precisely, which give very precise results data. So microbial community can be easily identified through these methods. Now, if you compare these methods like DGGE, TGGE, ARDRA and ARISA and T-RFLP, you find little variations, and every method has its own merit and demerit, and every method has its own advantage and applications. So, for example, in DGGE, TGGE, you have visual band patterns, right? In case of ARD and ARISA, we have the restriction fragment length patterns and variation of fragment of the enzymes used for the restriction digestion. Then we have automated processing takes place in RFLP through sequencer. Band sequencing possible. So you can take a band and sequence that band for further analysis, for mutations, for variations. Superior strains differentiation. Then quantitative measurement, right? Excellent for dominant taxa, right? Which is not in case of RFLP. Then you have a cost-effective analysis, right? In this case of ARDRA and ARISA, we have high throughput capabilities available. Then you have a simple methodology, right? Then in case of T-RFLP, we have high throughput analysis and statistical compatibility. So these are some applications and strength and things attached to these methods to understand the microbial diversity. Quickly we move to the which is the right method. So if you look when you're doing research in the lab, or as a master students or as a UG students, and look into the projects to identify the marker or to differentiate between the microbial diversity. So which method to be opted, right? Then we have to look into to observe first what is your hypothesis. So to look into the sample complexity, which sample you are working on. So consider the community, diversity level, and expected the species, the richness in your environmental sample. For example, if you are taking the wastewater treatment plant, you are taking the environmental plant, if you are taking the wastewater bodies, or if you are taking the rivers and ocean bodies, there you have to look into the chances of the diversity and abundance is more. So you have to look into what is the complexity of the genome, right? Then research objectives. What are the research objectives? So determine the need for the taxonomic identification versus the community fingerprinting analysis. So whether you are going for the taxonomic identification or for fingerprinting or the molecular analysis, right? So what kind of experiment you are preparing? What is your hypothesis, right? So that you have to look into before you pick up the method that you are working on. Then laboratory resources. So whether you have sequencer or not, you can't go for terminal fragment length polymorphism. You cannot go for sequencing. So look into do you have a PCR? If you have a PCR, you can do it. If you don't have a PCR, you can't do it, right? So look into the laboratory conditions and infrastructures and feasibility of this experiment, right? Accordingly, you have to choose the method. You should not go directly to the merit and demerit of the method, but to look into the infrastructures and the facility that you have. Budget constraint. Suppose if you go to T-RFLP or ARISA, the budget required is very high. It's a very expensive method. You have fluorescence probe, you have a primer to design, then you have a sequencing and so on. So all these things are important taking into consideration before you choose the method to be used, whether it is a DGGE, TGGE, ARDRA or ARISA or terminal length polymorphism, whatever method you are using for this marker identification, you have to look into the constraints, budget, equipment, facility, also skill, technical support, and what kind of a sample you are working on. What is the complexity of your samples, right? Whether it is a clinical sample, whether it is a environmental sample, whether it is some other diversified microbial samples, whether it is a gut microbiome, it is a vaginal microbiome, or it is an oral microbiome. So every microbiome has a different complexity. Accordingly, you have to choose the method. I think this is all. With this, I'm closing this talk today, and we'll meet again. Thank you very much.